children, welcome back to the class. This is Dr. Sweta Malani, your biology coach, and we are doing class 12, chapter 6, molecular basis of inheritance. This is our 10th lecture into the topic. In our previous one or two lectures, we were trying to find out how does the DNA replicate. So first I told you that the DNA replicates through a semi-conservative method which was proved by an experiment by Medalist and Stock. So what is the process of DNA replication? We were trying to find out. In my previous lecture, I gave you some interesting facts or important facts about DNA replication. Now it is time to find out the exact process through which it replicates, right? So let us try to find the same. How does the DNA replicate? It was Watson and Crick, as they gave their 3D model of DNA, the double helical model of DNA that was given by Watson and Crick. They suggested that the replication of DNA must be occurring in a set pattern. जब वॉटसन एंड क्रिक ने अपना डीएनए का डबल हेलिकल स्ट्रक्चर दिया था तो उसी वक्त उन्होंने बता दिया था कि डीएनए शायद ऐसे रेप्लिकेट करता होगा. They had already made a proposal that it must be replicating in a semi-conservative manner and in this method. However, whatever they said had to be experimentally proved. So what are the steps, exact steps that were found out later as to how the DNA replicates? So सबसे पहले while going through the step, we'll go one by one. So suppose this is my DNA and I need to make a copy of it. The first question that comes to our mind is, ये DNA कहाँ से replicate करना शुरू होगा? From where should I start replicating it? So normally what we feel is, from one end. Why don't we separate this end, make a new DNA across it, इस end से start करते हैं, ठीक है, इसके चारों तरफ इसका नया DNA बनाते हैं, नया DNA, then let us open little bit more, make the DNA open it more and like this maybe replicate. So ऐसा सोचने से तो ऐसा लगता है कि DNA replication must be starting from one point, that is one end. But studies reveal that no, in eukaryotes, since the DNA was too long, DNA बहुत ही ज़्यादा लंबा होता है, 2.2 मीटर लंबा DNA होता है. DNA replication एक end से शुरू नहीं होता, बल्कि multiple points से DNA replication हो सकता है, and उसमें भी there are fixed points. ऐसा नहीं है कि यहाँ से start हो गया, कभी यहाँ से, कभी यहाँ से, कभी यहाँ से, ऐसा नहीं. Whenever the DNA will replicate, it will replicate from fixed spots, and those fixed spots were called as ori or origin of replication. So before I show you this diagram, I want you to understand first from where will the replication start? From where will the replication start? So as I told you, the replication will start from fixed points on a eukaryotic DNA, and those fixed points are called as ori, also called as origin of replication. This means ori means origin of replication, right? ये origin of replication multiple पाए जाते हैं in eukaryotic DNA. Say if this is a eukaryotic DNA, I'm just making it ठीक, just like this. So here पहली बात तो कहीं से भी DNA replicate करना start नहीं करेगा, fixed point से करेगा, और इस fixed points को हम कहते हैं ori. And not one but many ori or origin of replication is found at least in case of eukaryotes. अभी मैं eukaryotes की बात कर रही हूँ, बाद में मैं prokaryotes की बात करूँगी, right? ये जो ori है, why is it that eukaryotes have multiple ori? You can ask me. This is one ori, this is one ori. Why? Say, I'm just giving you mathematically. It takes 1 minute for the DNA to replicate from here to here. यहाँ से यहाँ replicate करने में suppose 1 minute लगता है. तो डेफिनेटली यहाँ से हमें 1 लगेगा, प्लस यहाँ से 1, दैट मीन्स पूरे डीएनए को रेप्लिकेट करने के लिए 1, 2, 3, 4, दैट मीन्स 4 minutes will be required for replication of the entire DNA. Now suppose if I have multiple ori or origin of replication, from here to here it will require 1 minute. I said from here to here also it will require 1, from here to here also 1, here to here also 1. So by the time I have moved from here to here in 1 minute, तब तक यहाँ से यहाँ भी replicate हो चुका होगा, यहाँ से यहाँ भी, यहाँ से यहाँ भी, so that in a period of 1 minute, the entire DNA will be replicated. जब तक हम यहाँ से यहाँ करेंगे, तब तक यहाँ से भी replication start हो चुका है, वो यहाँ से यहाँ पहुँच चुका है, तब तक यहाँ से भी start हो चुका है, वो यहाँ से यहाँ पहुँच चुका है, वो यहाँ से यहाँ पहुँच चुका है. That means, see, the duration has been reduced many folds. Otherwise, it would have required 4 times more for this DNA to replicate had it just started from one end and continued till the other. Having multiple ori or origin of replication, it allows the DNA to replicate at a very faster rate. So this is what is about the eukaryote. Now you can ask me, ma'am, what about the prokaryotes? In prokaryote, the DNA is not as large as compared to eukaryotes or humans. Let me draw the diagram of a prokaryotic, that is a bacterial DNA. Now the bacterial DNA is very small as compared to a eukaryotic DNA. So in bacteria, there is only one ori or origin of replication. Again, this point is fixed. Any point cannot act as an ori. And here, there are two reasons why bacteria has only one ori or origin of replication. One, the DNA is short. First, here the DNA is very short. So it will not require as much time, so we do not require multiple ori. And second, in bacteria, the replication is bidirectional. That means on one hand replication will go from here, on one hand it will go from here, and at midpoint the entire replication will get over. So that is why there is only one ori or origin of replication in case of bacteria or in case of eukaryotes. So let us see it very quickly. I told you first, if you need to replicate your DNA, first you need to find out a place जहाँ से आप replication start करेंगे. उस जगह को हम कहते हैं origin of replication, जिसको short में we call it as ori, O-R-I, ori. You can see here in this DNA, there is this place where the origin will start, whom we have mentioned as ori or origin of DNA. In case of DNA replication, in case of prokaryotes, you can see here, they have a single ori or origin of replication. You can very clearly see it over here. There is a single ori or origin of replication, and the replication, this point is called as ori or origin of replication, and you can see that the replication is bidirectional. It moves either way. यहाँ से भी DNA देखो multiply हो रहा है. You can see it is opening here and it is multiplying. It is opening here. It is multiplying. That means the region in which the parental strands are separating. This is called a replication fork. What is replication fork? I will tell you later. Filhal, I just want to tell you, in prokaryotes there is only single ori or origin of replication, and DNA synthesis in prokaryotes is bidirectional. Coming to whenever the replication will start, at that point a bubble will be formed. Now what do you mean by this bubble? I just want to show you once again. See, this is my DNA. I've just made this DNA. Just see it closely. And this is the ori, the point from where the replication will start. So let me show this ori by this, blue, this silver ball. So this is the ori from where the replication will start. So this is the point from where the replication will start. So if it has to replicate, the DNA has to open so that it can multiply. So the moment it opens, it forms a bubble-like structure. You can see this. This open structure is called as bubble. So in case of prokaryotes, if you see this, there is a single bubble. Just look at the prokaryote. The DNA is opened here and there is a single bubble. But in eukaryotes, since there are many points of ori or origin of replication, here it will open, here it will open, here it will open. So it forms multiple bubbles, right? Because of multiple origin of replication. Now, once we have found out the ori or the origin of replication, कि भैया DNA कहाँ से replicate करेगा, start करेगा. Once we have found it out, all next what we need to do is open the DNA from that point. उस जगह से हमें DNA को खोलना जरूरी है. So let us see how will we open the DNA. I had kept two laces of two different colors just to show you a helically coiled DNA, just to give you the feel of a DNA. So it has opened. Let me do that very quickly once again. So okay, this is my thread. It is ready. It is a double coil, one black color, one red color. This is my ori or origin of replication. From here, the DNA first needs to uncoil. It needs to first open up. Now I need an enzyme to open it, right? I need an enzyme to open it. The name of this enzyme is helicase. I'm just going to draw it right now. And I want you to see this. So let me first tie it in one of my fingers and then I can show you better, more, just let me do that. Okay, so I need an enzyme called as helicase. This helicase will actually help the DNA open. Right? Now the moment I open the DNA and when I leave it, it will have a tendency to recoil once again. See, this is not very tightly done, or I could have shown you. Look at this. Do you see? When I open it, it opens, but when I leave, it has a tendency to coil back again. So what will happen? Look at here. What is happening? What is happening is, I have my DNA. This is how my DNA looks. This is my DNA. This DNA has a point of origin. This is called as OD, or the origin of replication. From this point, the DNA will open. Very good. The DNA will open like this. The DNA will open like this. For opening, first I need a point. This point is called as OD, or origin of replication. Second, I need an enzyme over here. The name of the enzyme which will help in opening it, this is called as helicase enzyme. Now the moment I open the DNA, it gets open. But when I leave it, what happens? It tends to coil back again. It will have a tendency to again coil back. ये जो हमने खोला है ना, this will again coil back. तो करें क्या? So we use a protein over here called as single strand binding protein, जिसको हम लोग short में SSB भी कहते हैं. SSB, also called as single strand binding protein. Single strand binding protein. And what does this protein do? It does not allow the DNA to recoil. जैसे हम लोग कपड़ा सुखाते हैं तो उसमें चिमटी लगाते हैं ना, so that the कपड़ा does not fly off. Similarly, it just puts itself like a clip, and it does not. Once you open the DNA, it will have a tendency to coil back again. It does not allow it to coil back again. This protein is called as single strand binding protein. So what we have done is, I'm going to show you once again. Third, I want to show you is that once you uncoil a DNA, at certain point it will get tight. Just like this. See, this is my DNA. If I am opening it, teeth, I need to put it in my teeth. Can you see? It has got tight here. इसके बाद it's not opening. It is difficult to open it at this point, right? So if it does not open beyond this, how will I open the remaining part of the DNA and make a duplicate of it, right? So here comes another enzyme, and that enzyme is called as topoisomerase. So there is another enzyme. I'm making that enzyme again by green color. This is called as topoisomerase enzyme. And what is the function of topoisomerase enzyme? It helps to relieve this tension that has been caused due to the coiling of the DNA. यहाँ पर ये क्या करता है, इस DNA को यहाँ और यहाँ एक बार काट देता है, so that the tension is released and so that the replication can further proceed. So I have told you about a group of enzymes and how the DNA replication starts. So let us see it once again. This is my DNA. I need to make a copy of the DNA. Where does it occur? Within the nucleus. When does it occur? During the S phase of interphase. Now, सबसे पहले I need to find out the exact spot from where the replication will start. That point is called as OD, or origin of replication, and this OD or origin of replication in eukaryotes we have only single OD or origin of replication, जबकि in case of in eukaryotes we have multiple OD or origin of replication, जबकि in prokaryotes we have just single OD or origin of replication, right? At this point the DNA uncoils. Because of the uncoiling you can see a typical V-shaped structure is produced. देखो, a typical V-shape, जब आप ऐसे खोलोगे तो एक V-shaped structure बनेगा. This looks like a fork. जो आप कांटा यूज करते हैं ना खाना वाना खाने के लिए. Say this is a fruit salad. It has fruits. You are using a fork to pick it up. Since it looks like a fork, we call it as replication fork. So we are learning a lot of new words. Let me just write them. This is called as the replication fork. Replication fork, right? So सबसे पहले क्या हो गया? We are finding the point from where the DNA replication needs to start. This is called as the OD or origin of replication. From this point the DNA will unwind. How will it unwind? It will unwind with the help of an enzyme called as helicase. ठीक है? Once it unwinds, it will have a tendency to rewind. So we will have a set of protein molecules called as SSB, or single strand binding protein. This will prevent it from getting recoiled. Because of the opening, a typical V-shaped structure is formed. This is called as the replication fork, and because of the uncoiling, here a tension is created. This tension is released by another enzyme known as topoisomerase. Right? So with that, the DNA replication has started. We have found out the point from where it will start. We have actually opened the two strands. We know that the two strands are anti-parallel. One is 3', 5', the other is 5', 3', and once it has opened, we have pinned it up with single-stranded binding protein, and then we have a topoisomerase which will help us release the tension and further unwind the DNA so that the DNA replication can move in a contiguous manner, right? So this is the introduction. So let us see what is there in the PowerPoint. So I told you first the DNA needs to be unwinded. Unwinding of the two DNA, it requires an enzyme. The name of the enzyme is helicase. Another name for helicase enzyme is unwindase enzyme. Since it helps to unwind the DNA, the enzyme is also called as unwindase enzyme. Helicase or unwindase enzyme. It helps to, unwind the DNA. You can see the DNA has been opened and it forms a typical Y-shaped structure. This will go on like this. If the DNA has opened like this, the replication will continue like this. Or if this is how the DNA's polarity is meant, see, so this makes a typical Y-shaped structure. This is called the replication fork. This takes place with the help of an enzyme called as helicase. Let us see those diagrams. And then it is stabilized. So this is the enzyme helicase, if you can see that. And this is the single strand binding. This is the enzyme helicase and this is the enzyme single strand binding protein, which allows it to remain separated, and this forms a typical V-shaped structure, and this is called as the replication fork. So let us see more diagrams to explain what has happened. This is helicase enzyme. And then you can see it, this is the single strand binding protein which is allowing it to remain single stranded. Now this has formed a typical V-shaped structure, and this structure is called as replication fork. So now that my DNA is unwound, it is ready to be replicated, what do I need to do? So let me do one thing, let me draw the DNA over here and then let us decide how it should go. So I have two strands of DNA, let me make one of the strands with red color. And let me make the other strand with another color, say a blue color, right? The red strand, say it has a polarity 3' and 5', and say the blue strand has a polarity 5' and 3', right? I have, with the help of enzyme helicase, I have opened it. With the help of enzyme single strand binding protein, I have already made it single stranded, and a typical replication fork has already appeared. See? Now that it has happened, I need to go ahead. So you know that here comes the role of enzyme, a DNA polymerase or DNA dependent DNA polymerase, which will do polymerization of the DNA. But at the same time, we are well aware that the polymerase enzyme will not start. There are two things, it will not start replication, it will continue. So we will require RNA primer, and secondly, the polymerization will be done in a fixed direction, that is in the direction 5', 3', and never in 3', 5'. So let us see, in this case there is no problem. First, what will we do? We will put a small primer molecule. This is the primer. Then this structure called as the DNA polymerase, DNA dependent polymerase, will attach over here. It will go on adding the nucleotides, right? It will go on adding the nucleotides, and this way the DNA replication will begin and it will move towards a replication fork. The newly synthesized DNA has a polarity 5', 3', and we are very well with it because my template DNA is already 3', 5'. Now let us look for the other strand. In the other strand, the problem is that here the polarity is 5', 3'. So replication needs to start from here. So I cannot start it from here, so I'll start it from here, because if this is 5', this is 3', so replication will start here. So here the primer will be added here, this primer here. Then the enzyme polymerase will come here also into action, and it will also allow the polymerization of the DNA, and thus a complementary DNA will be formed in the direction. The newly formed DNA is in the direction 5', 3'. So we are very well rated because it is going in an anti-parallel direction. Right? So you can see what I have taught you. Just see it once again, because this will get rubbed off. Some written material will come over here, and then we have to follow these diagrams. I told you one thing, please follow it step by step. The first step in DNA replication is ORI. ORI is also called as origin of replication. These are those points from where replication starts. ORI is multiple in case of eukaryotes, but in prokaryotes it is only one. Okay? At the point of ORI, the DNA unwinds with the help of an enzyme called as helicase, and this DNA is maintained in a single stranded structure with the help of another protein called as single strand binding protein. Once the DNA is uncoiled, it forms a typical Y-shaped structure which is also called as replication fork. Now what will happen is, well, I want the DNA to form the complementary strand. So the next enzyme that will come into the picture is DNA polymerase, also known as DNA dependent DNA polymerase. But the problem of DNA dependent DNA polymerase is, first it will polymerize only in 5' 3' direction, and the second problem is that it will never start replication, it can only continue it. So what we are trying to first figure out is the polarity. So of the two strands of DNA, the DNA which has been made in red color, it has a polarity, you can very well see, it has a polarity 3', 5', and the other has, the blue one has a polarity 5', 3'. So in this case, what happens, the primer is added towards the primer has to be added always toward the 3' end of the template strand. That strand on which the DNA is being made, that the original strand is called as the template strand. So here we add the primer. In the other one, the primer is added towards the replication fork. Then the polymerase enzyme comes into the picture. This is the polymerase enzyme. This polymerase enzyme will cause further polymerization of the DNA. That means it will add nucleotides with the help of the ATP supplied by the nucleotides themselves, and thus the DNA, the polymerization will go ahead. Once I have replicated till here, I need to further open the DNA. अब DNA को आगे खोलना है ताकि ये process आपका आगे बढ़ सके. But I told you that at this point a tension develops. And I also told you that this tension is released by another enzyme. You can see here, I have shown it. This enzyme is called as topoisomerase. It is also called as molecular scissor. It will actually cut the DNA temporarily over here, allow it to unwind, then attach it once again, thus relieving the DNA of the tension so that the process of DNA replication can continue further. Now we'll just have a look at the diagram which is over here before I actually change my page. So you can see this is the double-stranded DNA which is open. This has been opened with the help of the enzyme helicase. You can see over here, right? This is the enzyme helicase. Then it is maintained, the single strands are maintained with the help of enzyme single strand binding protein. Then we used RNA primer. You can see here we used primer. This primer, RNA primer, it is made with an enzyme known as primase. The primer is made by the enzyme primase. I don't know what I have done. Why have I added primer both ways? No, this is wrong. Only in this case the primer should have been added from this side. So this primer has been added with the help of the enzyme primase, and once the primer is made, then this enzyme DNA polymerase sets into action. For further uncoiling, I have another enzyme called as topoisomerase. Now let us move ahead. So unwinding of the two DNA, it is released, this tension is released with the help of enzyme topoisomerase. The unzipping causes a typical Y-shaped configuration which is also called as replication fork. So this is a diagram, I think this is not very clear, so we can omit it. Now let us come to this, how the enzyme topoisomerase works. So I told you this is how the DNA gets, it gets stuck over here, the tension develops here, then the enzyme topoisomerase comes into action. The purple color here is the enzyme topoisomerase. It will, this yellow one is the topoisomerase. It will cut the DNA at 2 points. Then what will happen? It will open, it will get unwind like this, and then it will again make it join, thus it releases the tension. So this is just a diagrammatic representation to show how topoisomerase releases the stress. Now the next diagram shows how the replication fork goes. So I hope you can see this clearly over here. So this is two strands of DNA. One of the two strands of DNA has a polarity, you can see 3' and 5', and the other has a polarity 5' and 3'. Now that DNA which has a polarity 3', 5', there the polymerase enzyme, that this is up till here it's the primer, then the polymerase enzyme will act on, and the polymerase will continue the DNA replication in this direction. But the other strand who has a polarity 5', 3', the replication will actually start from the point of the replication fork, and from here first the primer will be added, and then the enzyme DNA polymerase will allow the replication to take place in this direction. So this is, let us see. So limits of DNA polymerase 3. Actually you must be seeing that here it is always coming polymerase 3, polymerase 3. What are these polymerase 1 and 2 then? And what is this polymerase 3? I will tell you in due course. DNA dependent DNA polymerase, that it is of 3 types. Polymerase 1, polymerase 2, polymerase 3. Only polymerase 3 it helps in polymerization of DNA. What does polymerase 1 and 2 do? I will tell you later. So it is just saying that what is the limitation? It can not initiate replication, but it can only continue. So please just watch here how things work, a little animated version. सबसे पहले what will happen here, this is the 3 prime 5 prime, so the enzyme primase will attach. Because of the enzyme primase, a small RNA primer is formed at this point. I told you what is a primer. It is a short strand of DNA of known nucleotide, and the primer which we use is RNA primer. Why? Because it is made up of ATC. It is not made up of ATCG, rather it is made up of AUCG. You know that in RNA we have uracil in place of thymine. Right, so first this is my parent DNA. We had the primase enzyme which synthesized RNA. The moment the RNA was synthesized, now what will happen is the enzyme DNA polymerase will attach, and it will ensure continuity of this RNA into the complementary DNA. Now if you look at the other strand, what will happen is the primase will be formed here, but here in this case the direction will not be this. The direction is not going to be this, it is going to be that. So the primase is attached, then you can see here red colored primer is formed, and then the polymerase enzyme will further help us formation of the new strand. Actually what happens is in this strand, as the DNA goes on uncoiling, the DNA elongation will be continuous. But in this case, let me just show you first on the board, then you will understand this diagram. Say this is my DNA, right? Say this is my DNA, I uncoil my DNA to this amount. And what happened was, in this case the DNA was formed like this, and in this case the DNA was formed like this. Now this DNA further uncoils, right? Now it further uncoils. Now when it further uncoils, this one which has proceeded till here, this will not have a problem. This will further move like this. But in this case, you know, it started from this point, it went here. Now another primer needs to be added here. Once again a primer needs to be added here, and once again the DNA will be synthesized like this. That means in this strand the DNA will be made in short pieces. I will explain you further, and you know better. I think I should actually explain you once again, I think little properly. Say this is my DNA. Let me make it little. This is, please notice 1, 2, 3, 4, 5. I have made this like this. This is my DNA, it has opened like this. In one of the strands whose polarity is 3 prime, 5 prime, this is the primer attached here, this is the primer attached here, and the newly formed DNA will go like this, and it will go like this. Now as the DNA will unwind, see I am unwinding the DNA, now it is like this and like this. I have opened it further, see I have opened it further. So what happened was, here we had the primer, here we had the primer, and the new DNA was formed like this here and like this here. Once DNA is further uncoiled, for this it is not problem, it will move straight ahead. But for this it is problem because it has come to an end over here. So once again a new primer needs to be made attached here, and once again the polymerase which was formed over here, this polymerase which had reached here, in this case the polymerase reached here, it further continued to reach here. But in this case the polymerase which started from here has reached here, it has to be taken out. Once again it will come here to the primer, and once again a new DNA will be formed like this. Right? I will show you this on a bigger board right now. So this is what it is showing. This is another primer, you can see. Once again a primer has been formed, then again another. That means in one of the DNA strands whose polarity is 3 prime 5 prime, there the new DNA is formed continuously, while the other one whose polarity is 5 prime 3 prime, the DNA is formed in short segments, and these short segments are called as Okazaki fragments. This will continue till we go till the end of the DNA. So let us see. First we have a primer. You can see that this primer is built up by an enzyme polymerase. This serves as a starter for the sequence of DNA polymerase 3. So you can see this. So the use of primer, it is given here once again in the notes. The primer, it actually, the synthesis of new strand, it begins on this molecule, you can see over here. This is called the primer. A primer is a short chain of RNA formed by polymerization of nucleotide UCANG with the help of enzyme primase. So in one of the strand the primer has been formed here, in the other it has been formed here. This is the enzyme primase which helps in the formation of this primer. Once the primer has been formed, then the DNA polymerase enzyme will attach here and allow the polymerization in the required direction. So let us see. In the other place you can see here, let us look at the strand, how the DNA polymerization takes place. Once the polymerase enzyme has been added, it will add on the nucleotides. It will continue to add the nucleotide and thus the newly synthesized DNA will continue to move in the forward direction. In one of the two strands, synthesis of two strands, you will see, in one of the two strands, as the enzyme DNA polymerase attaches, here you can see this. That strand which has a polarity 3' and 5', that strand, here the polymerase enzyme will move in a continuous manner, and as the DNA uncoils, it will continue to make the DNA in a continuous manner. But that strand where the DNA polymerization is being done in the opposite direction, here every time you want to make a DNA, you have to attach a primase. You have to attach a primer. See, the polymerase enzyme went from here to here. Now again if I open it, then again a primer has to be made here. This polymerase will come here. Again the DNA molecule will move ahead. That means in one of the strand, you can see the DNA is formed in a continuous manner. This is called as the leading strand. Why it is called as the leading strand? Because here DNA formation is continuous. So no one is stopping. So obviously वो फटाफट कर रहा है, वो करता ही चला जा रहा है. This is called as the lagging strand. Why? Because I have to stop every now and then. ये वाला जो DNA है, इसमें तो ऐसे continuous जा रहा है. इस वाले में क्या है? तो इतना बना, फिर खुला, फिर again I need to put a primer, again I need to put polymerase, again I need to form a short fragment, then खुला, फिर again I need to put a polymer. So यहाँ बीच-बीच रुक रहे हैं ना, तो ये थोड़ा पीछे हो जाता है इस strand से. ये फटाफट आगे बढ़ रहा है, so we call it as leading strand. इसको थोड़ा वक्त लगता है, so we call it as the lagging strand, and it forms DNA in short fragments. These short fragments are called as Okazaki fragments, right? So this is on the other strand of DNA. The DNA is formed in smaller fragments, and these smaller fragments, they are actually called as Okazaki fragments. So let us see. So this way, let us see the summary. How the DNA replication. First, we have the enzyme helicase. Then we have the enzyme single strand binding protein. This is the direction in which the replication fork will grow. So this is a V-shaped structure called as replication fork, and the DNA is supposed to continue in this direction. So I'm showing you the polarity. This is the 3' end, this is the 5' end. Accordingly, that is the 5' end and this is the 3' end. In this, what happens? The polymerization will occur in this direction, that is 5', 3' direction. And in this, the polymerization cannot occur in this direction. See, it's putting up a wrong. So here, this will occur in this direction. In that case, it will occur in opposite direction. You can see it very well over here. So here, it will be formed in smaller fragments. These smaller fragments are called as Okazaki fragment. In this, it will be a continuous strand. So this is called as the leading strand, and this is called as the lagging strand. Once these segments are formed, later these segments, we have multiple RNA primers over here, because at the beginning, every time we need a RNA primer, right? We have multiple RNA primers. So these RNA primers are later removed, right? And these short strands are joined with the help of an enzyme called as ligase. Once this ligase enzyme is used, now this DNA also becomes continuous, but it lags behind in speed as compared to this one. So this strand is called as lagging strand, and since this occurs in a continuous manner, so obviously it takes place fast, and this strand is called as your leading strand, right? So let us see some more animations here. These fragments are later joined by enzyme ligase. These fragments are called as Okazaki fragment after the name of the scientist Okazaki. Now DNA ligase, you can see it will come into picture, and this will allow these short pieces to join together. This strand is called as the leading strand because here the DNA synthesis is continuous. That strand is called as the lagging strand because here the DNA synthesis is in small fragments. It takes larger time, and then they also need to be joined again with the help of the enzyme ligase. ठीक, the lagging strand, you can see here it's written the lagging strand is called lagging strand because it is made up of smaller fragments called as Okazaki fragment, and these fragments are later joined by ligase enzyme which act as welders. They weld these smaller pieces and they make a continuous DNA in this strand as well. So let us have the summary once again. First we have the enzyme DNA helicase. It helps in unwinding of the DNA. Then we have the single strand binding protein. This helps to keep the DNA single stranded. Right? Then we have a typical Y-shaped structure coming up. This is called as a replication fork. Now we have the polarity. You can see one of them is 3' 5', the other is 5' 3'. Now the DNA polymerase will come into action. This DNA polymerase will help in polymerization only in 5', 3' direction, as you can see. So it will polymerize only in this direction. So what will happen is, in this strand, it's a complete repetition. The DNA form will be in a continuous manner. While in the other one, you cannot start replication from this end. You have to start replication from this end. That is from the point of replication 4. Here, the DNA is made in smaller fragments. These smaller fragments are called as Okazaki fragment after the name of the scientist. Later, these fragments, they will be joined with the help of an enzyme called as DNA ligase. Right? You can see this. Now this strand, since it is formed in a continuous manner, this is called as a leading strand, right? And the other strand is called as the lagging strand. The lagging strand with the smaller fragments, Okazaki fragments, they are joined by the enzyme ligase. Now I told you that in DNA, the polymerization is at a very high speed, approximately 2000 molecules per second, 2000 nucleotides per second. Now that's too fast. I said that they do it with a lot of accuracy, but still 1 in 10,000 they do a mistake. 10,000 में एक बार mistake हो जाता है. I told you that the polymerization of the bases occurs at a very fast speed. Say if this DNA has nucleotide sequence A, T, C, G, C, 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 G, C. Now the enzyme polymerase will put here T, A, G, C, G, 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 C, G, and this polymerization which the DNA polymerase enzyme will do, it will do at the rate of 2000 nucleotides per second. Now, doing it at such a high speed, there can be some mistake, and the mistake actually occurs 1 in a 10,000. One mistake may happen in the base pairing. So we must have a provision of proofreading. Now, what is proofreading? When your teacher, she sets questions for your exams, there can be a probability that there is a mistake in the question paper. So once the question paper has been typed, right, it goes back to the teacher for a proofreading. That means they go through the question paper once again to see if there are no errors. Once the proofreading has been done okay, only then the question papers, they go into print. Similarly, once the DNA polymerase 3 moves simultaneously, इसके पीछे we have another polymerase called as either DNA polymerase 1 or DNA polymerase 2. It also moves at the back to check whether the polymerization is being done right or wrong. If any place there is a mistake, for example, if here it was C, it was supposed to be G, but by mistake the polymerase puts here instead of G, it puts say maybe T. So this polymerase 1 and 2, this polymerase will move ahead. This will move ahead. And what did it do? It placed T here instead of G. As this polymerase comes, it will see, oh, there's a mistake. It will remove this T, it will replace it by G, and it will rectify all the mistakes. So there is a provision of proofreading in the case of DNA replication as well. DNA polymerase initially makes about 1 in 10,000 base pairing errors. 1 in 10,000 it can have an error. So it has enzymes which proofread and correct these mistakes. Okay, the new error rate has been 1 in 1 billion. Earlier it was thought that this mistake is 1 in 10,000, but the new error rate that we have found out is 1 in a billion, but that 1 in a billion should also not happen. So we have polymerase 1 which does proofreading. It repairs the mismatched bases, it removes the abnormal bases, and it reduces the error rate from 1 in 10,000 to 1 in 100 millions. That means 1 in 1 billion. Right? The initial error rate is, that was a mistake. The error rate that can be performed by DNA polymerase 3. DNA polymerase 3 से 1 in 10,000 का error होता है. But because of the proofreading, this error reduces from 1 in 10,000 to 1 in 100 million to join together. This strand is called as the leading strand because here the DNA synthesis is continuous. That strand is called as the lagging strand because here the DNA synthesis is in small fragments. It takes larger time, and then they also need to be joined again with the help of the enzyme ligase. Okay, the lagging strand, you can see here it's written the lagging strand is called lagging strand because it is made up of smaller fragments called as Okazaki fragment, and these fragments are later joined by ligase enzyme which act as welders. They weld these smaller pieces and they make a continuous DNA in this strand as well. So let us have the summary once again. First we have the enzyme DNA helicase. It helps in unwinding of the DNA. Then we have the single strand binding protein. This helps to keep the DNA single stranded. Right? Then we have a typical Y-shaped structure coming up. This is called as a replication fork. Now we have the polarity. You can see one of them is 3' 5', the other is 5' 3'. Now the DNA polymerase will come into action. This DNA polymerase will help in polymerization only in 5', 3' direction, as you can see. So it will polymerize only in this direction. So what will happen is, in this strand, it's a complete repetition. The DNA form will be in a continuous manner. While in the other one, you cannot start replication from this end. You have to start replication from this end. That is from the point of replication 4. Here, the DNA is made in smaller fragments. These smaller fragments are called as Okazaki fragment after the name of the scientist. Later, these fragments they will be joined with the help of an enzyme called as DNA ligase. Right? You can see this. Now this strand, since it is formed in a continuous manner, this is called as a leading strand, right? And the other strand is called as the lagging strand. The lagging strand with the smaller fragments, Okazaki fragments, they are joined by the enzyme ligase. Now I told you that in DNA, the polymerization is at a very high speed, approximately 2000 molecules per second, 2000 nucleotides per second. Now that's too fast. I said that they do it with a lot of accuracy, but still 1 in 10,000 they do a mistake. 10,000 में एक बार mistake हो जाता है. I told you that the polymerization of the bases occurs at a very fast speed. Say if this DNA has nucleotide sequence A, T, C, G, C, 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 G, C. Now the enzyme polymerase will put here T, A, G, C, G, 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 C, G, and this polymerization which the DNA polymerase enzyme will do, it will do at the rate of 2000 nucleotides per second. Now, doing it at such a high speed, there can be some mistake, and the mistake actually occurs 1 in a 10,000. One mistake may happen in the base pairing. So we must have a provision of proofreading. Now, what is proofreading? When your teacher, she sets questions for your exams, there can be a probability that there is a mistake in the question paper. So once the question paper has been typed, right, it goes back to the teacher for a proofreading. That means they go through the question paper once again to see if there are no errors. Once the proofreading has been done okay, only then the question papers they go into print. Similarly, once the DNA polymerase 3 moves simultaneously, इसके पीछे we have another polymerase called as either DNA polymerase 1 or DNA polymerase 2. It also moves at the back to check whether the polymerization is being done right or wrong. If any place there is a mistake, for example, if here it was C, it was supposed to be G, but by mistake the polymerase puts here instead of G, it puts say maybe T. So this polymerase 1 and 2, this polymerase will move ahead. This will move ahead. And what did it do? It placed T here instead of G. As this polymerase comes, it will see, oh, there's a mistake. It will remove this T, it will replace it by G, and it will rectify all the mistakes. So there is a provision of proofreading in the case of DNA replication as well. DNA polymerase initially makes about 1 in 10,000 base pairing errors. 1 in 10,000 it can have an error. So it has enzymes which proofread and correct these mistakes. And after this proofreading, see what happens. After this proofreading, the mistakes, they come down to 1 in 1 billion. Imagine. You understood what it's given. It's saying that, first we have the DNA polymerase 3. This DNA polymerase 3 does the polymerization task. Okay. First we have DNA polymerase 3. So let me write it. The DNA polymerase 3, it does polymerization at the rate of 2000 base pairs per second. This leads to approximately error in the count of 1 in 10,000. 10,000 में एक error लग जाता है. पीछे कौन आता है? DNA polymerase 1 and 2. Now DNA polymerase 1 and 2, they come and check the mistakes and they rectify it. As a result, the mistake drops down to 1 in a billion. From 1 in 10,000, so it comes to 1 in billion. Okay? So just see this, it's just showing that here there was a mistake. Okay? So what happens? The polymerase enzyme comes, it removes it, it replaces it by the correct one, and this is how the repair takes place. So DNA polymerase 1, it proofreads, it reads it, पीछे आता है, read करता है, it corrects the mistakes, mismatches are repaired, every damage is repaired so that the error is reduced from 1 in 10,000 to 1 in 100 million bases. So I am just giving you an overview of the types of polymerase. This is DNA polymerase 3. This is the main builder. Here I have written it is 1000 base per second, but in your NCERT it says it polymerizes at the rate of 2000 bases per second. So it is the main builder polymerase. You can see this. So this is the main polymerase which does the building work. Then we have your polymerase 3 के बाद we have polymerase 1 and 2. They are basically meant for editing, repair and primer remover. The primers which are formed, that also needs to be removed ना, then with ligase it will get joined. मैं क्या बोल रही हूँ कि जब आप replication करते हो तो आप क्या ऐड करते हो? आप ऐड करते हो ना primers. Say this is my DNA, so we add primers here. These primers also needs to be removed. So removal of these primers, then editing, repair, this is done by polymerase 1 and 2, and the repair rate is approximately 20 bases per second. At this rate they do the repair work. So with that we have completed DNA replication. Let me quickly make you all revise it and let you know how and what you will write if it comes as a 3 or 5 mark question in the board exam or the school exam. Then we will go through the NCERT to see what is written there before we conclude today's lecture. So we are studying DNA replication. The first step is, that is about ORI. About ORI, 1. These are the places where the replication starts. 2. The number of ORI in eukaryote. Then what it is, you have to write. Then the number of ORI in eukaryote is many. But in prokaryote it is single. It is this place from where the DNA replication can start. Once you have identified the ORI, you use the enzyme helicase. This enzyme helicase helps you unwind the DNA. So this will help you unwind the DNA. Once it is unwound, the single strand binding protein will help them remain stabilized so that they again do not get coiled once again. Then will come the role of this results in the formation of a typical Y-shaped structure, and this is called as replication fork. Okay? So once you have written that replication fork, now you will tell about the role of primers. With the help of an enzyme primase, primer is formed. In the that DNA template, which is 3' 5', it is done towards the origin, but whose polarity is 5', 3', it is done towards the replication fork. This primer, so next is formation of primers. This is done with the help of enzyme primase, and this is a short strand of DNA of known sequence and where it is placed. Once this is done, we will have the role of DNA polymerase enzyme. The DNA polymerase enzyme 3 will get attached over here, and this will lead to extension of the DNA strands, right? Then we will have the role of topoisomerase enzyme. With the help of enzyme topoisomerase, the DNA is further uncoiled. In one of the strands, the DNA replication will be continuous. This strand is called as the leading strand. In the other, it will take in short fragments. The fragments are called as Okazaki fragment, and the strand is known as the lagging strand. This will continue till it reaches the end. Thus, we will have two DNA formed, both of them semi-conservative, one parental and one newly synthesized DNA. Then this polymerase, it does a mistake 1 in 10,000 since the speed is high, so there may be a mistake 1 in 10,000 in pairing the base pairs. This is rectified with the help of certain different polymerase which act as for proofreading. They go behind it, they proofread it, and they reduce the mistake from 1 to 10,000 to 1 to 1 billion. Okay? So these are the various steps of DNA replication. All these steps and all these enzymes are not given in your NCERT. So in your board exam, please be careful in not mentioning those points which are not in your NCERT. For example, this point, single strand binding protein is not there in your NCERT. Then polymerase 3 and polymerase 1 and 2 is not there in your NCERT. So whatever is not in your NCERT, you will not mention in your exam. Further, I have a doubt, we will go through NCERT, we will come to know if even helicase is there, because a popular question that comes in the board exam is name 2 enzymes that are used in the process of DNA replication, and the 2 enzymes which are clearly in your NCERT, one of them is your enzyme, DNA polymerase, one of the enzyme, and the other enzyme is the DNA ligase, which helps to join these fragments. The fragment is named as Okazaki fragment. Again, this is not there in your NCERT. So let us have a look at the NCERT to see what is found over there. Open page 106 of your NCERT. You can see, besides it is saying DNA dependent DNA polymerase, many other enzymes are required to complete the process of replication with accuracy. For long DNA molecules, since the 2 DNA cannot be separated in the entire length, you cannot separate both the DNA first. कि चलो पहले दोनों को अलग कर देते हैं. You cannot do it like this, कि this is 2 DNA, first let us separate both of them and then I form a new DNA. No, you cannot do that. You will separate little bit and duplicate simultaneously. थोड़ा separate किया, duplicate किया, फिर थोड़ा separate किया, duplicate किया, फिर थोड़ा separate किया, duplicate किया. This is how it happens. Since it happens in only small areas, so small openings occur where the replication takes place. This is called as replication fork. DNA dependent DNA polymerase polymerizes only in one direction, 5 prime 3 prime. Thus in one of the strand the DNA replication is continuous, in the other one whose polarity is 5 prime 3 prime it is discontinuous. The discontinuous strands, they are actually joined with the help of an enzyme ligase. The DNA polymerase on their own does not initiate. You can see the DNA polymerase cannot initiate, so they require randomly at any place. This DNA polymerase cannot do DNA replication from any place, so you need fixed places for them. These places are called as ORI or origin of replication. Okay? And this ORI or origin of replication has been of great use to us in our DNA technology, that is in biotechnology. What this is, only this line you will not understand right now, you will understand in the chapter biotechnology. So in your book you can see it is so preliminary. It is just saying that the DNA is open, it's not even giving the name of the enzyme, and then it is saying it opens at fixed points called as helicase, a replication focus form, and then the DNA polymerase makes the strand, in one it makes continuous, in one it makes discontinuous, and the discontinuous strands are joined through ligase. Other than that, it has not mentioned anything else. And there is a diagram also. This is a very important diagram both in board and medical. So this is the diagram you have to make in case you have, it comes in the board exam to draw the diagram of DNA replication. So you can see this is a very silly or a very primitive type of diagram. In this diagram you can see it has made a double stranded DNA, one with polarity 5 prime 3 prime, the other is in 3 prime 5 prime. In 3 prime 5 prime the DNA is formed continuous, and in the other one it is formed discontinuous, and these discontinuous is basically joined by DNA ligase. Only this much is given in your NCERT, right? So with this the process of DNA replication is, I am sure, very clear to you right now. अब आपको अच्छे समझ में आ गया होगा. So please complete this portion as well, because we have completed the first function of DNA, that is the replication. The next function of DNA is to express itself, what we call as protein synthesis, which occurs in 2 steps, transcription and translation. So in next class we are going to start with transcription. Till then, please continue studying, and please put your comments and put your like or dislike, whatever you feel. Share it with your friends, subscribe if you are new. Until then, as I always say, please take care of yourself, but keep studying as well. Till then, thank you and goodbye.